This is day 5 after fertilization, which means that the embryo now is in the uterine cavity, and the stage is called the blastocyst. The blastocyst that we're looking at here has a layer of cells that surround it. We call it the trophoblasts, this layer. And we have a mass of cells on one side. We call it the embryoblast. Embryoblast means the precursor to the embryo. The trophoblast means the nutritional layer, or the nutritional cells, or the cells that are going to be supporting that embryo. There's something missing here. There used to be the zona pellucida outside here, the shell of that embryo, and now it's lost. It broke down, which means that the trophoblast can get in contact now with the wall of the uterus, the endometrium. Specifically, this area where the inner cell mass is, the trophoblast here can get attached to the epithelium that lines the uterine cavity. So, if you recall, fertilization happened in the fallopian tube. And throughout those 5 days, the fertilized egg started division and moved, or was pushed, all the way to the uterine cavity. And as it was being pushed, it developed through several stages until it reached the blastocyst. Blastocyst here has a cavity. We call that the primary yolk sac. That's the old name. The more correct name is the blastocele, or the blastocystic cavity. Has the inner cell mass and the trophoblast. The trophoblast will get in contact with the endometrium and attach to the endometrium in anticipation for implantation around day 6. Then we move to the next stage, which is the implantation itself. Happens around day 7, where the trophoblast that you see here, that got in contact with the endometrium that you see here. This is the endometrium. And you can tell that the trophoblast now have a second layer. These arms are actually a syncytium, or a continuum of cells with no cell boundaries between them, no cell membrane. The cytoplasm is continuous. All of these nuclei here are of the same syncytium. And this is a highly invasive structure that allows the blastocyst to be buried into the uterine wall. As you can see here, it's burrowing through the uterine wall. Again, the blastocyst will have the trophoblast, which are now 2 layers. The cytotrophoblast are these cells here. The in situ trophoblasts are out here. They are the ones that are invading the wall. And you have the inner cell mass. And before I leave this stage, something that was not shown in this image, these surface cells here will start showing different behavior. They will form a layer, which is the earliest of the precursors, cell layers of the embryo. We call it the hypoderm, which is the primitive endoderm. It will not become the endoderm. It will contribute to the endoderm cell layer, and we will discuss it later. But it is the precursor to that layer, or one of the precursors to that layer. We will focus from now on the inner cell mass. We will discuss how the embryo itself develops, instead of focusing on or describing all the events that happen around it. But before we do that, let's insert that structure deeper into the wall, make it completely implanted around day 7, day 8. Probably this structure here, this in situ trophoblast, is so extensive. We're talking about probably day 10, where it's now invading through really deep through the wall, even cutting through glands and blood vessels, and the blood vessels will leak their blood into that syncytium, forming pools of arterial and venous blood, which basically is the precursor for the placenta. Now, if you're inside the uterine cavity, which is here, you don't see that structure at all. You just see that plug, mucosal plug that blocks that entry wound. Where is the inner cell mass? The inner cell mass is now differentiated into two layers, only two layers. Instead of a mass of cells, it became a two-layer disc. Remember, this is a two-dimensional cut in a three-dimensional structure. So what I'm pointing to or circling here is a two-layer disc. If I flip it from above and look at it from above, it will be like a disc. And the two layers are the hypoderm, okay, which is the layer below here, and the epiderm, okay, which is the layer above here. And I want to tell you, perhaps you have heard these terms before, the ectoderm, the mesoderm, and the endoderm, which will form all the tissues that we have in our body. Just to eliminate the confusion, most of these three layers are actually coming from that epiderm, not the hypoderm. The hypoderm contributes to the endoderm that we have in our bodies, but not to all of it. So most of the tissues are actually coming from the epiderm, and in the next stage, or actually in the next week, it will be of development, it will be three layers instead of two. What about these cavities? This cavity here used to be the primary yolk sac or the blastocystic cavity. Now it's called the primary umbilical vesicle. This layer here, which actually is a cavity that developed within the inner cell mass to isolate those two layers and from the rest of the inner cell mass, and having a fluid in that cavity, which we will call the amnion, and the cavity will be called the amniotic cavity. What about the cavity around it? The cavity around it actually developed within the trophoblasts, within the cytotrophoblast layer, and that is the one that will become the gestational sac. And it will be very important for the development to come. But from this point on, we will only focus on what happens in those two layers, that disc that will develop into the human body.